So guys, I am Kamran Khan. I'm basically the final year student now in final semester, and I'm doing environmental engineering from National University of Sciences and Technology, Islamabad. And that's it. And
Okay.
now I recently, okay, let me share another thing with you. Now, actually, I'm active on climate change studies and on especially the green technology, stuff like that. And I founded a new student chapter, international student chapter in Pakistan. Its name is American Academy of Environmental Engineers and Scientists, and it will start working like within a week or 2 weeks, and I'm hopeful that I will, start like recognizing our environmental engineers and making them linked with international organizations.
Okay.
Yeah.
Anything else you wanted to share about?
That's,
That's okay.
that,
Okay.
yeah.
Hi, this is Saad Khan, and I'm doing a specialization in inorganic chemistry. I'm here in Karachi, studying in a Federal Urdu University, Karachi campus, and I'm still working on my research paper for synthesis of biodiesel from different waste. What our goal is, what our slogan is, from waste to gold. Right now, we are facing lots of trouble in our climate change, just like Zika virus and other viruses are just destroying other nations in Africa, in subcontinent, and also in China. So for that purpose, my teachers and my other members of my class are working on it, and they got a good result. Because I can't share all the result with you because this is top secret for them. If I reveal, they will dump on my face. Okay. Okay, let's come. You may start with your presentation because all I have to do is to talk and talk and talk on
Okay.
biodiesel. It's a renewable fuel for transportation. Basically, here
Yeah.
in Pakistan, government is not as much taking part in research
Mhm.
work like for biodiesel, bioethanol from the agriculture sectors. Even our farmers are not educated that how much land they have to use for farming and how much land they have to use for the forestation. Over here in Pakistan, less than 50% of our area in is not included in forest. Deforestation is common over here in Pakistan. Do you want to share some point, Kamran?
Yes. Thank you. Yeah, it's a very interesting topic. Yeah, it's to make biofuel from like food waste and other organic waste is it's a modern concern nowadays. Especially in Pakistan now, we are facing the energy crisis, and we have also some economic concerns. We it's expensive like for Pakistani people to import the fuel from other countries. So the biofuel is basically the domestically like originated and generated fuel. And my studies is based on to produce biofuel from algae. It's basically the third generation generated biofuel, and I will share my studies on that. Um, I have prepared some, some slides for that to share with you people.
At the bottom of your screen, there's a green box with an arrow pointing up. If you hit that, you can share your screen.
Yeah, okay. Okay. Can you see it now?
Yeah, we can see it.
Okay. So basically, it is my final year project, and I was working on this project, that how we can fully utilize the algae for different purposes. So my basic concern was to extract carotenoids and to produce biofuel from the residual biomass, which was you know which remain as in residual material after extracting the lipids, proteins, and
Sorry, Kamran, can you put a full screen, please?
Is it fine now?
Yeah.
Yes, thank you so much.
Okay. So I was sharing with you people, like I was doing some research where to extract some carotenoids. Carotenoids are basically protein in nature, and my second phase of my final year project was to produce biofuel from the residual biomass, which is in which is like in residual mass, which left after extracting carotenoids, proteins, and lipids. So basically, the carotenoids are fat-soluble pigments which are synthesized by plants, algae, and photosynthetic bacteria, and they have the antioxidant properties. So it can, protect us from, cancerous cells because of the antioxidant produced, because it produce radicals. And these are some of the health application of the carotenoids. And now on Saad concern, like biogas, as we all are well familiar about the benefits of the biofuels. Like it's economical, it's cheaper as compared to the conventional conventional fuels. And it's also less harmful as compared to conventional biofuels, and it is easy and environmental friendly also. So for it, so how I was producing the biogas from the algae, like algae, as we know that to produce biogas from any material, we need some organics. So like algae can be widely producible. Like it just need some natural habitat. It needs the natural condition, favorable conditions. It needs some favorable temperature, light, and densities. It needs the optimum nutrients. It needs a high concentration of nitrogen and phosphorus. So if we can just throw some algal species in a limited amount of water, having optimum nutrients, it start producing algae, and algae growth is exponential. So we can further collect the algae. We can collect the algae by screening them, and then we harvest them in different bioreactors. What I had used in my final year project was PSP, or it was photostatic bioreactor, and actually I harvest algal biomass, and after extracting the dry mass, I basically produce the biogas by giving optimum conditions. So this was the problem statement. We have some limited research on algal uses, which restricted how we can fully utilize in terms of health and economic feasibility. Our aim was to develop and transform renewable biomass resources into commercially viable high-performance bioproduct. and biofuels through target research and development. Okay. So this was my literature review, and this is basically the methodology. So first of all, like I cultured the given algae, and what I had chosen was the Hematococcus bovalis. It is basically one of the freshwater grown algae, and I um, grew it in suspension phase. So and it's it's like additional benefit it is like it can also treat the water quality, like it can treat the wastewater by extracting the nutrients from it. And then I go for the cultivation phase, and I cultivated algae in the port substrate bioreactor, and then we checked the water quality in which the algae was grown. The water quality was much better. I had seen a difference of like like you can say like 40 to 60% of the difference. Like it extracted not the heavy metals, but especially the nutrients like nitrogen and phosphorus. And then we harvested it, and we then extracted it some keratinites and the biomass which was left in the end, and then we uh, give some we gave some inoculum to degrade the given biomass, and after that in a closed container they start producing the biogas, and then we collect it in the the collection chambers. Basically these are some the growth parameters for the algae growth. We have given some some optimum parameters. These parameters were taken by a literature review, like carbon dioxide should be in 0.03 to 5%. With increase in the carbon dioxide, it basically change the acidic, it change the pH value of the like medium in which the algae is growing. And the light intensity is basically the 750 photons, and you can also say it as a micromoles per meter square second. And algal biomass, which was left in the end after extracting keratinoids, we treated that algal biomass by thermal hydrolysis, like by giving them some heat to break the cell wall of the algae, and to let them biodegrade easily. And we had also given some like anaerobic conditions within the container by producing a nitrogen gas into it, inert gas into it, by removing the oxygen from it, and then ultimately you get the biogas from it. It's very complex process. Actually, I was restricted just to extract the keratinoids, and now I'm working on the, to extract a biofuel from it. And my research was only restricted towards the extraction of the keratinoids. But after that, my supervisor, insisted me and suggested me to go towards the the second phase, which is basically the production of the biofuel. And I'm still also working on it, and it means this literature review. Actually, it's a third generation study and on internet, especially on LG, there is less work available, like how we can fully utilize the residual biomass. So these were some of presentations, and okay. Okay, now it's your turn. Saad, I know I was so boring.
Okay, Kamran, you have done a lot of work because you are a research student.
Yeah,
Basically, what I'm doing here in my university, I'm volunteering as a researcher. I'm not a authorized and a official researcher at my department. I'm just volunteering for my information that what happened when we react something and what we get, why don't we get some things. So in this way, I can start my work. So basically, about talking about the biodiesel and a biofuel. Getting bioethanol is not an easy task. From crops and maize, we can get it. But for getting bioethanol, we have to cultivate some land to grow maize. Maize is also a food crop. But while we are taking maize as a bioethanol, so that contain shortage of food in some areas of our country, like here in Pakistan. I have my friends and I done this thing, and all the farmers are saying, "What you have done? Where all our maize? Where are our maize?" So they are so much worried about the income because people over here in Pakistan are not so many people are not economically established like us.
yes.
Okay, so what we have done over here in our depart, we are collecting different kinds of waste and different kinds of bones, like the bones of goat, totters of goats, and the horns of buffalo, cow, sheep, and goat also. So what we have to do first, we are just making bioethanol from these kinds of waste, and also we are using these things into as a fertilizer also. So for making a biodiesel for this thing, so we are just calcinating it. Do you know the term calcination? What is it, Kamran?
Calcination is basically no. I'm
Okay,
just not aware of
it's
it.
something like a sister of a pyrolysis.
Okay, okay,
Just like a pyrolysis, but we have a different kind of calcinator that is heating in the absence of oxygen, but we are just moving it to make our nanoparticles. What we are doing, we just have to calcinate our waste, just like we have if we are having the
so you are
goat
saying
bone.
like you are by like degrading the waste, like okay, the bone ash. You are like
No, 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 that's
bone
not
ash.
ash. That's
Like,
not ash. We also have done experiment that by using a calcinator and also combustion in the open air.
yeah,
So in open air we can get a ash, but the result with the ash is not as suitable for getting a biodiesel in a good quantity or in a useful manner. For this we are just only able to do calcination for the goats and totters. What we are doing, we are just calcinating over to it about an three and a half hour for 600 degrees Celsius to 900 degrees Celsius. Sometimes it goes to 3.5 hour to 4 hour because it depends on our sample. So it's like 2 days to 3 days, and then we are making a nanoparticles of all our waste, all our calcinated samples, and then we are reacting with some kind of a biochar to get it. And to get it. Basically, what we are using over here, we are just making a reaction by using the sunlight. We are having a big lenses in our depart for from our teacher. They gifted us for our research work, so we are using those lenses. that can concentrate the light on it on a sample, and the photons of tiny and little energy particles of light are just erecting on it, and then we are getting a yield. But it takes 3 or more than a 5 hour to get a desired result. Without using any electricity, we are just using sunlight to get the biodiesel. After that, the reaction when the reaction is done, so we are not confirm that our biodiesel is prepared or not. So for that, we are taking a TLC, thin layer chromatography of our sample. Do you know what is a chromatography is?
Yeah, yeah.
Okay, okay.
It's a good.
So, so we are using TLC to guess that either the diesel from these samples from the goats, or horns from of the cow, or horns of the goats, getting a diesel in a good quantity or not. Sometimes the reactions goes in a very bad way, because when we are doing the TLC, so we have to make a mixture of a toluene and chloroform, and using other chemicals mix as a mixture to make a TLC run in the TLC container. So sometimes the water particles comes in a toluene, and then a whole reaction will just
Like
go in a
for
wrong.
identification purpose, sorry for the interruption. Like
Yeah.
for the identification process, you mentioned that you are using, the chromatography. No, am I right?
Yeah, yeah, we are using chromatography, thin layer chromatography, TLC.
Okay, it's great. Did you use some another procedures like, the high performance liquid chromatography, and it's a very advanced process, to
Yeah,
check
you're
the
talking about the
amount.
HPLC, na?
Yeah, yeah.
Yeah.
It's,
So basically, first initially test is done by in this way. For other purpose, to getting a to know our sample, so we are moving towards the atomic absorption. We are doing this in this way, that that collecting the samples, we are just washing it with the deionized water, and then filter twice with the filter paper of a 0.G4 size filter paper, and then we are just put samples on the adsorption. Then we get the result, either what elements and what metals are present, and how our biodiesel can we get in a most
yeah.
in a what can we say, yeah, here is the pictures of the lenses that is Sarah is sharing with you guys. Here is what we have done. We are having a over here is a hot plate that is only for the purpose of the steering, because we are over here in a laboratory, we don't have enough shaker that we can go outside in the sun. So we are using a hot plate for the steering, and then we are using the lamps. All these things are designed by my colleagues and me also. We are designing this to the last hole here to process, to done our reaction. So what over here they are doing it, we have a flask. In it we are just putting the some 2 grams or 1 gram of our sample to collecting our biodiesel for this. Over here, can Sarah, can you move the picture? Yeah, over here we are just taking it, we are concentrating the lights on the bottom of the flask, and meanwhile we are just taking the temperature. We are knowing the temperature, what temperature is in the flask, by using the IR gun. That is a thermometer that is used infrared radiation to get the temperature. Sarah, can you move for me please, next picture also? Yeah, over here in this is the last stage of our reaction. What we have done over here, just it's about a 4 hour reaction. After that we are putting all the reacting in the flask into a washed glass, and making it dry for a 15 to 20 minutes, and then we are taking a sample to the laboratory for the TLC. After that, we run the TLC in a ultraviolet light, to knowing that how much yield we can get from our reactions. And we are using also iodine for this, to knowing that how far our TLC can run, how far the biodiesel can run, and by this way we are getting the RF factor, retardation factor from this. and this is how we can collect our diesel. But commercially we are not getting enough from the government of Pakistan over here. Kamran, do you want to say something about the government of Pakistan? Are they taking any action for biodiesel or not?
Yeah, actually, especially when I was when I choose this project, to extract and to produce biofuel from algae. It's a very easy and cheaper process as compared to conventional procedure to dig to extract the oil from underground basins. It's a very complicated process. It's need the manpower. It needs the high technology. It needs technology for the further refining procedures. But in the LG case, it needs just a land. I can share a picture with you. I was just looking into it. Okay. Um, Sarah, could you please share my screen over there? Okay, you can see the ponds where the algae is growing. You
Yeah.
just need to screen out the algae from over there, and the the process which needs some attention is the harvesting. You can do it by the different procedure. What I had mentioned to you people, what I had shared with you people is the PSPR. Okay. Okay, basically the BSBR, the photo stationary bioreactor, on bio membrane and any membrane of any different material, we just put that screen biomass on it, and we remove water from that biomass to make it dry, and after 24 hours and any given time, we make it dry, and after drying procedure, harvesting procedure, we produce some inoculum in it, and inoculum is basically the sludge, and having different microorganisms to biodegrade the dry mass of algae. So to biodegrade the algae, we had given some anaerobic conditions in a in a container. So yeah, it's a very easy process, but it needs the government attention, to commercialize the process, and to you know and to improve this methodology to industrial scale.
Okay.
And I just want to share some of my studies about suspension phase. Suspension phase is that phase which is the initial phase. In this phase, when we put the algal species in a water having a medium, a nutrient medium in which the algae start growing, and the nutrient medium which we had chosen for effective growth was Bohr's Basin medium. Meanwhile, we kept observing the different strategies of algal cells under microscope, showing the proper growth of algae. And the protocol was to autoclave distilled water and BBN. And the range of light intensity we have prepared for fast growth was 750 micromole photons, what I had shared with you people earlier. And you know, just to avoid the contamination, we wash and disinfect all the equipments and the apparatus before use, to avoid any contamination. Because when you are using, the algae for hull, for like consumption purposes, like for like if you are concerned with just to extract the carbonites, then you have to avoid the contamination. And if you are just concerned to produce the biofuel, then the contamination is not a big concern. And you can easily just go for the harvesting procedures and harvest the algae, making it, converting it into a dry uh, biomass, and then introducing the inoculum in it in a closed container, giving them anaerobic conditions, and it can easily, you know, change that biomass into gas. One of the challenge is to optimize the procedure is the treatment of the algae. What the term treatment, especially in biofuel, is like to enhance the production rate. To enhance the production rate, we give some heat to erupt the cell wall of algae, and we also introduce some chemicals into the containers to increase the productivity rate of the algae. Productivity rate of the biofuel, I mean. So the biggest challenge is to collect the produced biogas. And when algae was biodegraded, it produced different types of gases like methane. It produced the methane, it produced the carbon dioxide, it produced the hydrogen. So the biggest challenge after biodegradation of algae is how we can separate the different types of gases. And what's our concern? If someone is interested only in hydrogen, then he needs to follow the different protocol for it. Uh, you know, for hydrogen, they can adopt the different absorption mechanisms, and for the methane, like similarly, the different absorption media can also be used to separate the different types of gases. One like for qualitative analysis, we can for the quantitative analysis we can use HPLC. HPLC is basically the high performance liquid chromatography, and in the high performance liquid chromatography, we can check like like how much amount of a specific gas, specific ratio of gas is present in a specific sample. So this is what I am working on this project, and still I'm refining my procedures and my working approach to make it more feasible for commercial purposes. And but like why like industries are not following that procedures, because we have limited research, and like and especially the stakeholders and the investors are confused that can we adopt that procedures for for commercial production of biogas. Like what I had mentioned with you people is like the collection of the biogas is the biggest challenge. So to convert, uh, are you listening me? Okay, so that's fine.
Yeah.
Okay. It's a very complicated process. Like I'm also, confused in my mind. Like what I need to share with you people, because what
Just
I
you
did.
have to just what you have to do. Just you have to summarize all of your stuff, what comes in your mind, and what the common people can understand it.
Yeah. Okay. That's what I am trying. I think so. You understand the basic procedure. Like if you want to, um, grow algae, just take the sample of the algae from anywhere and put it into the the wastewater, in any water which have some nutrients in it. And algae is very hungry to uptake the nutrients from the water. And if you have the water sample having enough nutrients like nitrogen and phosphorus and another accessory, the nutrients, and it start growing and its growth is exponential. After that, when you have grown enough, the
Can we
algae.
put the algae into a fresh water in which contains fish and other marine animals? Can we put algae in it on it?
Because in the fresh water we don't have enough nutrients, and we need some favorable conditions for algae, and there are some algae which is the freshwater producing freshwater grown algae, but over there they need some specific conditions, like specific temperature. It needs specific nutrients, but and it needs the nutrients are the basic requirements to grow algae, because like plants, if you want to grow the plant, it need nutrients which is basically extracted from the soil. The same cases with
So either we can blend the biodiesel with the diesel to run our car and motorbikes or not.
its efficiency is quite productive. What our seniors did with the algae, they produce biodiesel from the algae, and the quantity of the biodiesel was optimum enough, and they checked the efficiency by the HPLC and by the different procedures. And I'm not familiar about the efficiency procedure, like how you can check the efficiency,
Okay.
but the purity of that sample was quite inspiring, and they even to check the biodiesel, they burn that sample and check the flame. Like it was a physical
Checking,
efficiency.
not a chemical checking. Yeah.
Yeah, like for the to be honest, I'm not familiar about the
Okay, it
efficiency
doesn't
process.
matter, but you know lots of things about it.
Yeah, and it's basically to grow algae in water and having optimum nutrients is basically the suspension phase, and to make it dry, it is basically the harvesting phase. In the harvesting phase, when you extract the water from algae and make it dry, it is basically the second phase. And after the second phase, you have to produce the inoculum in it. Inoculum is basically the sludge. Sludge
Okay.
has the different types of microorganisms, and basically it depends like what type of conditions you are providing to the algae. If you are providing the aerobic conditions, you have to provide the aerobic microorganisms, because microorganisms needs sustaining conditions. If you put anaerobic microorganism into the aerobic conditions, then it's very difficult for the that specific microorganisms to sustain that environment. So what you have to do is like to biodegrade that dry biomass. You need to select some anaerobic microorganisms, um, and put that microorganisms into a closed container. and then it start biodegrading. Like it's a very simple procedure, and it's and
So
any
either, sorry for interruption. So
yeah.
either any member of government can contact to you for the production of the biodiesel or not.
Actually, our president of Chamber of Commerce contacted me. It was to produce, like carotenoids. It is, the one of the component of the algae. Algae has the fats, proteins, lipids, and many other components. In my first phase of my final year project, I extracted carotenoids and I completed it, and I showed the results. And the president of the Chamber of Commerce was inspired, and he requested me to keep it moving. Actually, in Pakistan, in pharmaceutical industries, they import different types of chemicals and different types of salts from um, different countries. And in Pakistan, there is no industries which can produce their own salts for like pharmaceutical purposes. So
Yeah, because
basically,
over here in
yeah,
Pakistan, all the pharmaceutical companies are getting their raw material
yeah,
from other countries. What the main
yeah,
focus we have that we have to make our raw materials over here in Pakistan to lower the cost of the
yeah,
medicines here in Pakistan. Yeah, what
yeah.
I discussed with the so many people of that are connected with the pharmaceutical company, they're just saying, "Oh, just shut your mouth. This cannot be possible." What I think it can be possible. Either we can strengthen ourselves to make a raw
Mhm.
material over here in Pakistan.
Like we can still, see the significance of algae. We can produce some salts from it. We can extract the carotenoids from it. We can even get the lipids, and by separating the lipids, we can produce the biofuel. Like we have the organics, and it's very easy to grow algae, and we can get many other benefits, additional benefits by growing the algae. It's too environmental friendly. It's in the favor of the environment, and the biofuel which is generated and
Okay,
produced.
can sorry for interruption. Can
Yeah,
we give the 10 minutes to other people to
sure,
ask
sure.
questions from us?
Yeah, sure.
Okay. Okay, guys, you can ask anything if you want with us.
Ines, why don't you go first?
Yeah, thank you. I was waiting.
Okay.
My question basically is for Cameron. Cameron,
Yes,
Okay.
are you both from Pakistan, right?
yes,
Yeah,
we
we
are.
are both from Pakistan.
Yeah, that's great because I was just I didn't see. I mean, just I just see. I was here just from the beginning of the presentation, the PowerPoint. But I see that I'm working on Algae as well in another aspect. But I see that you just, I mean, enter to the method of extraction and dash PLC and go on. But the first step, I would like to know what I mean species of algae are you working on? That's very important. You are
Yes.
talking about algae on general. There is a millions of algae. Are you working on red, brown, or red algae? And also, what method of harvesting are you doing? Where do you bring your samples of algae? Before talking about the extraction is like the last step of the process. That's very
Okay.
important. What species are you working
Okay.
on, and where do you bring it?
Okay, I got your question. It's a very interesting question. And actually, it took more than 3 months to specify on which algae we need to work on it. And I have chosen two species, which was both of them was fresh water grown algae. And the first one was Chlorella vulgaris, and the second was the Hematococcus pluralis. So both of them are the freshwater grown algae. And what was the reason behind that? That we had chosen those two algal species because our FIP project was split into two phases. The first phase was to extract the keratinite, and the second phase was to, produce biofuel from the left uh, residual biomass at the end after the extraction. So like in Haematococcus cruvalis, like there is in one cell there is 80% of the astaxanthin, the keratinite present in it. Like very economical to extract the keratinites from Haematococcus because it have a high, like you can say the protein mass, the the carotenoid, which was our concern. So that's why we, chosen the Haematococcus cruvalis. And after that, the Florida vulgaris, it also have, the beta um, carotene in it. It is another carotenoid which is also have some, the beneficial properties in health industry and in the pharmaceutical industries. Um, these were the reason that we had chosen all of these species. So from where we got these algal species, actually we screened them by taking, the algal species. Um, the sample was taken by one of our, uh, you know, co-supervisor, which was from another university, and we requested them that we need this kind of algal species, and we didn't put some efforts in screening purposes, and they actually did screening for us, and they gave us, the isolated form of of that algae, and actually we didn't isolate it that, and we didn't put some, efforts in the screening procedures. And for Haematococcus cruvalis, we actually import that species from Norway, and it was just to save our time. But I'm now interested, to work on the species in screening purposes, and to work on national grown algal species. I'm hopeful I answered your question.
Yeah, yeah, very interesting. But I just want to, do you harvest seasonally? I mean, every season or monthly, every month, or how you do? Because we know that the algae, algal species, their physiology is different, from every
Yeah,
season. I'm not working on the extraction,
yeah,
but I know that the physiology sometimes I'm in the coast, sometimes in a season in winter, I find
mhm,
this spicy. For example, now the spicy, the Ginidium or or Cystozera or whatever.
yeah,
I
yeah,
find it in a state, but in another season it changed. The morphology changed, the physiology changed, everything changed. I mean, it, do you take this in consideration in your research? I don't know if you are doing a PhD research or another stuff.
yeah. It's again another interesting, thing, and what I did was to check, like, is there any physiological change, especially the material component of the algae. And we noticed that, like, to grow algae, there are some stages. The first stage is, the lag, and then the stationary phase, and then, the regression phase. So yeah, like if you uh, don't provide them optimum condition, then the algal species like goes directly into the regression phase, and it dies out. So you need some, the optimum conditions and mother conditions to grow them viably and feasibly, and and to get the optimum growth. So yeah, you're right. Like there are a number of species which are going in their mother condition. Like some of them are freshwater born algal species. Some of them are saline water born algal species. Some of them are like you can say like and some of the species are red. Some of them are brown algal species. Some of them are the blue, green algal species. So these species need mother conditions to grow, and mother conditions are basically the light intensities, the nutrient availability, and and um, you can say the saline condition for them. So every species are dependent on their mother conditions. Yes. If you have any questions, please. Did I answer your question?
Okay, nobody has a question. I have another one.
Yeah. Okay.
No,
It's a very interesting topic. Yeah.
it's very serious because I know what you're doing because it demands a bit of knowledge, to interact and you know what I mean. Someone from the field that can understand what you're doing. Because it's, he was talking as well about the physical-chemical analysis of water, right?
Yeah. Right.
You did some. I heard the nitrogenous parameter and phosphorus parameter.
Can you repeat that thing again? What you are trying to say?
You did some phosphorus and nitrogenous parameters.
Yeah. Yeah.
I mean, what's the purpose of this analysis in the relation with the exploitation of algae?
Um, if I got you, correctly, what I got you is basically you are trying to ask about the parameters in the suspension phase, like why we are concerned to give them optimal conditions.
No. I'm telling you, you make some physical-chemical analysis of sea water, right?
Yeah.
What's the purpose of this analysis in relation with the exploitation of algae or the extraction of algae?
Okay. It's very interesting, like if you get the algal species which was grown and which was taken from the freshwater samples, so you have to give them such properties, some such parameters, like some mother condition for that. And actually, I'm not expert to grow, the saline water grown algal species, but like it's a natural process. It's a natural mechanism. If something is like addicted, and if something is adopted to some conditions, you are bound to give them some conditions to that. And I am I am again sharing with you people, like I'm not expert in growing saline water, grown
Because
algal
because
species.
uh,
Yeah.
because because I'm gonna tell you because why I was asking from where do you bring the samples? Because if you extract from them some substances or you want to make something, and then you have to
Yeah.
bring them from a safe area, which means
Yeah.
a clean area.
Yeah.
For example, I make some physical-chemical analysis in seawater. I make some nitrogenous phosphorus parameters, but this is for an ecological purpose, for an ecological, objective. I do this, for example, to evaluate if this area is polluted. For example, in an area in the Atlantic coast,
Yeah.
Morocco, I found there is a high level of the full stage of development. I found there is a high level in this area. For example, I can't, for example, sample from here, you know, because it's very, I can say it's closed. I mean, you know, this
Yes.
parameter can help us. The purpose is like to see if this, you know, this area where I'm working on, and also I'm interested also to see the method that you use for sampling. And
The sampling have different types of procedures. You can Google them. If you want to extract the saline water sample, what precautionary measures should be taken to take that sample. But for fresh water, there are some precautionary measures. If you are taking a sample from sea, from river, and there are some uh, like evaluatory procedures. What I had mentioned with you people, and especially with you, that for we started our FYP after getting that sample, and we were not involved in spraying that species, and we were not involved and not worked on getting that species like uh, by sampling, by getting that sample from any respected area. So we just starting our project by taking that sample from our course supervisor, which was from another university, and we started working on it. Our main concern was how we can optimize the growth conditions. Like if it's a natural condition, its cell growth is 2030, in 24 hours, how we can make it 60 to 70 by giving them optimal conditions. And the optimal condition was taken by literature review. Like if we have taken the freshwater algae, which is the Hematococcus or the Chlorella, how we can optimize the growth especially in the suspension phase. So that's what was our concern, and we were not involved in the streaking phase, in the straining phase, and the isolating the different types of algal species. It's a very complicated process. You need to be a bit more careful. So actually, I did study the straining procedures, the streaking procedures, but I was just trying to focus to extract them, extract the carotenoid from them, and what could be the favorable procedure and optimal procedure to extract them. And we use different organic chemicals for the extraction purposes. We will just uh, sh- uh, like shrinking our FIP project to just some specific goods.
okay, thank you.
Okay, you're most welcome.
All right, do we have any last questions? I think our presenters have to leave
Yeah.
because it's almost time to break the fast.
Yes,
Yeah.
Yay!
really. We are in hurry. Let them have the
So
off, and
let's
my
not
mother
keep
is
them
calling me. Come on the
waiting.
table, on the table.
Yeah, we
Actually,
still
my
need
friend
seven
is.
hours here. We still need 7 hours here in Morocco.
Oh,
7 hours.
oh, okay.
Okay. Thank you guys for your time and listening our presentation. It's
Thank
really
you for sharing your
complicated.
knowledge.
Yeah, actually.
It was very interesting. Thank you so much.
Yeah, thank you very much.
Thank you,
Okay,
everyone. Our next call, just so you know, is going to be on May 30th at, let me check. at 4:30 p.m. GMT, and it's going to be on climate change and global governance by
Okay.
Tayeb and Ngozo. It's going to be a really interesting presentation as well. Thank you so much, Cameron and Saad, for your discussion about biofuels today. It was really interesting to learn about it. I'd never really talked about biofuels to this extent, so I really appreciate you sharing your research with us.
Okay. Thank you.
All right.
Thank you very
Thank
much.
you. Enjoy your dinner.
Goodbye.
Bye.
Bye.
Okay, thank you.